Welcome back to Biotech Simplified. Today we will discuss about Introduction to Bioinformatics. So what is bioinformatics? In simple language, we can say that bioinformatics is a combination of biology as well as IT. So a combination of biology and IT is known as bioinformatics. So the mother of bioinformatics is Margaret Dayhoff. So we can see that bioinformatics is an interdisciplinary field of science because in bioinformatics there are several branches of science which have been combined to form the branch bioinformatics. So in bioinformatics there is computer science, biology, chemistry, biochemistry, statistics, mathematics and engineering. So all these fields together forms the branch of science known as bioinformatics. So first we discussed what is bioinformatics. Next, let us discuss what are its application. So the first application is that bioinformatics is being used in biological data storage and retrieval. So the biological data is being stored in the form of database. So what is database? Database is a collection of biological data. So it is an orderly collection of biological data. So after which a biological data is stored in a database, it can be retrieved, that is, it can be accessed from the database. And for access, we use certain tools known as retrieval tools. So this is a main application of bioinformatics. That is, bioinformatics is being used for creating databases as well as for creating retrieval tools. A database are of different types. A database can be classified into several based on the data type, data source, and based on a data source, we can divide a data into two types, which is primary data as well as secondary data. So based on a database design, also we can divide it into two types, which is rational and object-oriented. So the last type of database is composite databases. So as we discussed earlier, a biological database can be of different types. First type of a biological database is bibliographic database. That is, it is a database which contains informations. That is, it is a literature database. The next type of database is taxonomic database. That is, from the name itself, we can identify what type of information it contains. It contains the information regarding classification. So, the third type of database is nucleic acid databases. That is, it contains the information about DNA. The next type of database is genomic database. That is, it contains the information about gene level. Next type of database is protein databases. So, the protein database consists the information about proteins. Next type of database is protein family, domains and functional sites. That is, it contains information regarding the classification of protein as well as the identification of domains. The next type and the last type of databases is enzyme or metabolic pathways. So it contains the information about metabolic pathways. So these are the common types of biological databases: bibliographic databases, taxonomic databases, nucleic acid databases, genomic database, protein databases, protein family domains and functional sites databases, and enzymes or metabolic pathway databases. The most common retrieval tools that are being used in bioinformatics are DBGT, Entrix, and SRS. So these are the most commonly used retrieval tools in bioinformatics. So now let us discuss the second application of bioinformatics, which is sequence analysis. This means that it is the comparison of biological sequences. So, if the comparison is between two sequences, it is known as pairwise alignment. So, if the comparison is between two sequences, it is known as pairwise alignment. And if the comparison is between more than two sequences, it is known as multiple sequence alignment. So, it is pairwise alignment and multiple sequence alignment. So, example of pairwise alignment is BLAST, and example of multiple alignment is CLUSTAL W. So this is another application of bioinformatics. So as we discussed earlier, a pairwise alignment compares between two sequences, whereas a multiple sequence alignment compares between more than two sequences. So the difference between pairwise and multiple sequence alignment is that multiple sequences are being compared in multiple sequence alignment, whereas in pairwise alignment only two sequences are being compared. Pairwise alignment are of two types: local alignment and global alignment. Local alignment compares the query sequence with certain parts of the target sequence. That is, only certain part of the target sequence is being compared. Whereas in global alignment, the entire sequence, that is, from the first sequence to the last sequence, is being compared. So this is the difference between local alignment and global alignment. That is, in local alignment, only certain parts of the alignment is being compared, whereas in local alignment, the entire sequence of the target is being taken into account. Another difference between local and global alignment is that in local alignment, only similarities are being taken into account, whereas in global alignment, both similarities and dissimilarities are being compared. In local alignment, Smith-Waterman algorithm is being used, whereas in global alignment, Needleman-Wunsch algorithm is being used. So, the differences between local and global alignment is that in global alignment, the full sequence is compared, whereas in local alignment, only parts of the sequence is being compared. And in global alignment, the algorithm used is Needleman-Wunsch algorithm, whereas in local alignment, the algorithm used is Smith-Waterman algorithm. So the table cells are given any score in global alignment, whereas in local alignment, the table cells are never score given below 0. So these are the basic difference between global alignment and local alignment. So global and local alignment are types of pairwise alignments. Now let us discuss the third application of bioinformatics, which is phylogenetic analysis. So what is phylogenetic analysis? It is the study of evolutionary relationships. So through phylogenetic analysis, we can create a phylogenetic tree, and this phylogenetic tree can be developed using phylogenetic tool. So By phylogenetic tool, a phylogenetic tree can be constructed, and through the phylogenetic tree, phylogenetic analysis can be carried out. So the tools for phylogenetic analysis are Phylip and Mega. So these are the two common tools that are being used in phylogenetics analysis. So this is a phylogenetic tree. The parts of a phylogenetic tree are root, branch, taxon, node, and clade. So these are the parts of a phylogenetic tree. Now we will discuss the fourth application of bioinformatics, which is CAD. The expansion of CAD is computer-aided drug discovery or design. CAD is a recent field, and this field is a combination of bioinformatics and aligned fields like chemoinformatics and chemistry. So, CAD is computer-aided drug discovery. So, a drug discovery is a complex and a risky treasure hunt. So, in the treasure hunt, we look forward in finding a molecule which is non-toxic. So, in the end of the treasure hunt, we look forward in finding a molecule which is non-toxic in effect. As we discussed earlier, CAD is a treasure hunt, and in this treasure hunt, we find a drug which is non-toxic and is suitable for our demands. So when the treasure hunt starts, we have 10,000 to 20,000 components with us, and during the several steps of CAD, the number of components left with us is decreasing, and in the final step, we are left with just one drug. Clinical and preclinical steps are done in this one drug, and finally, this one drug is being marketed. So the whole process takes 10 to 15 years, and it is a very expensive method. Hope the basics of bioinformatics is clear. For more such videos, do subscribe to our channel Biotech Simplified. Please do like, share and comment.